So now it's time to talk about DNA replication. DNA replication is the process where DNA copies itself and makes more DNA, which is quite an achievement in that it does it sort of ultimately all by itself. I mean, it makes proteins. DNA directs the production of proteins, which in turn end up replicating the DNA that's making the proteins. But we'll talk more about that in a few minutes. It turns out DNA replication is kind of a complicated process. It requires maybe 20 different enzymes. Some of them are called helicases. Helicases basically move along the double helix, and at origins, and that's what I'm trying to label over here, they'll actually pop open the DNA and actually allow replication to take place. An area that helicase is working, and so helicase is actually moving this way, it's right here. This is called a replication fork. This right here and this here make up the fork, and there's two forks in here, here and there. Two replication forks connected here. This is called a bubble. So this whole thing is called a bubble, but the left side here is called a fork, and the right side here is called a fork. It turns out the most important enzyme that operates here is called DNA polymerase. It is DNA polymerase that bonds new monomer nucleotides to the nucleotides of the existing strand that's being copied. Something that's important to point out that I'm going to point out, on and on and on. Biologically, always, DNA is read by enzymes in the 3' to 5' direction. That's the only way it's read. So it says here nucleotides of the template strand are being copied using the DNA in the 3' to 5' direction. In reality, it's picky, but DNA polymerase actually grabs what are called nucleoside triphosphates. If you remember from the energy and chemical unit, we talked about ATP. ATP, adenosine triphosphate, is actually a nucleoside triphosphate. It's adenine, but it's a triphosphate. And what DNA polymerase does is it grabs the triphosphate, it cuts the two phosphates off, two of the three phosphates off, and then puts it into the DNA to replicate. You'll see more about that in just a minute. Chromosomes, as it turns out, are linear, whereas bacterial genomes are circular. In chromosomes, DNA replication begins at many places called origins, and that's what I'm showing just in one section over here. DNA is antiparallel, which creates the problem replication cannot proceed in the same direction on both strands or on the opposite sides of DNA. As it turns out, when a bubble opens up, sections of the two forks are what are called lagging replication sections, and other sections of the two forks are called leading replication segments. A researcher named Reiji Okazaki actually discovered the short segments here and here on the lagging side of DNA replication. I'm going to animate this for you in just a minute, but there is another enzyme called DNA ligase that's important to replication. Ligase is also important in biotechnology, along with polymerase. Ligase actually is what is responsible for banding the little Okazaki fragments that are made on the lagging side of replication. Another enzyme is called topoisomerase. Topoisomerase actually deals with the unwinding of the DNA. When a bubble opens, this is basically kind of flat. And as the DNA is replicated, it's replicated back into a helix that's twisted, but these sections here are not flat. So when the bubble opens, these start to actually wind up. They super wind. So topoisomerase actually cuts little nicks in one side of the DNA, and that actually allows it to unwind its tension, and then it puts the little nick back together. It'll do that on both sides. So it's called the unwinding enzyme. And then lastly, primases actually are responsible for starting replication on the DNA. They actually lay down a little section of, I hate to say it, RNA, which is then used to actually start DNA replication along. Matthew Meselson and Franklin Stahl performed a landmark experiment in 1958 where they used radioactive nitrogen in DNA to show that DNA is replicated in what is called a semi-conservative fashion. By semi-conservative, what we mean is that the original DNA molecule is preserved in the copy process. What it is it is unzipped, and then new DNA sides are produced to fill in the missing sides after the unzipping. Let me see if I can show you a simple step-by-step animation of what that looks like. Let's see, can we find it? Here we go, bubbles. So this is, let's say, a piece of DNA. Here's the 5' end sticking out there. The bottom down here would have a 5' end sticking out here. And this is a piece of DNA that is equivalent to like what would represent a chromosome. So it's one chromosome before DNA replication. Now, there are multiple places on the chromosome that represent origins. So at origins, helicases will actually unzip the DNA and then create 2 forks and go in opposite directions. And at each fork, the DNA opens up. Now, since DNA polymerase can only go in the 3' and 5' direction, at this little spot in the fork, let's see if we can't draw on this. At this little spot, this is the origin here and here. At this little spot on the top of this bubble, DNA polymerase will literally just zip down and follow helicase. Helicase is right there unzipping the DNA, and it'll go all the way down and just keep unzipping the DNA through here. Meanwhile, these sections can't do the same thing, because when DNA polymerase attaches here, it can't go to the right, it has to go to the left. So the DNA here, the DNA polymerase attaches here and it moves forward, and then it hits replicated DNA and then it stops. Then another DNA polymerase will attach here. It has to go to the left, so it goes up and it hits replicated DNA and then it detaches. A similar scenario is happening on the bottom, but in the opposite direction. When DNA polymerase attaches here, it just smoothly moves to the right, following helicase to the right. On the left here, DNA polymerases, when they attach, they have to go to the right. So let's erase those pieces now. Now, as the replication goes, what you see is I got to get the right tool selected. There we go. The DNA molecule actually opens up wider and wider, so you can see more. These are called Okazaki fragments, okay? And the DNA polymerase is moving in the direction of the arrows. So when the helicase moves down here and opens up new DNA, polymerase will attach down here and go up. Meanwhile, down on the bottom, the helicase is literally right behind the DNA polymerase here on the bottom strand is right behind the helicase, replicating smoothly on the bottom side of the fork. This section, let's go ahead and draw on this. This section right here, this is called a fork. This is a fork here. This is a fork here. This is a fork here. And this is a fork here. So we have 4 forks essentially shown in the picture, and the forks actually replicate kind of opposite to one another within the bubble, within a bubble. So let's erase and mention here that this is leading replication here, leading replication here, lagging replication with Okazaki fragments, lagging replication with Okazaki fragments. Over here, leading, lagging, leading, lagging. Okay? Now, to continue with the animation, let's go ahead and choose the cursor. Now I have to click here. Okay, so now if we go one step further, the bubbles are even larger, and eventually the bubbles run into each other. Now you'll notice we have these little sections, and they really don't wait this long to get repaired, but you see the Okazaki fragments are not bonded together on the lagging sides of the replication. This is where ligase has got to come in and put those things together, and that's what it does. And the last step of this process is ligase coming in and mending the sides where the bonds are missing between the Okazaki fragments. Now we have 2 pieces of DNA that are absolutely identical because the DNA polymerases have replaced all the missing nucleotides with the correct nucleotides. So let's go back to the animations that are in the outline, and let's take a look at a detailed animation here. So this is a graphic here. You see in the graphic on the blue triangle here is helicase, unzipping the DNA. This is a DNA polymerase here, and you see this arrow here? It's zipping to the right side because it's moving in the 3' to 5' direction on the existing strand. By the way, the new DNA is being made in the 5' DNA, the two sides are anti-parallel. On the top, DNA polymerase will attach here and move to the left. It already attached here, moved to the left. It attached here, moved to the left. And then this is ligase putting together the little Okazaki fragments. This is an Okazaki fragment right here. This is another one here, and it's being bonded to this one there. So this is topoisomerase here, which is the unwinding enzyme that's allowing this to be flat temporarily while the helicase and the DNA polymerases and the primases and the ligases and stuff are all working in here. Now, this animation is rather simple, so let's go to this one and I'll kind of step through it. This is a linear chromosome. I'm not showing a true replication bubble in this animation. Let's make it a little bit bigger. I'm showing replication from one end. The key is helicase. It's popping the DNA open. And you'll notice on the bottom, the paintbrush, which is DNA polymerase, is simply attaching, and it's going to put a nucleotide in. In reality, this adenine nucleotide is actually a nucleoside triphosphate. The DNA polymerase takes the two phosphates that would have been sticking on here, it takes them off and throws them out. So the polymerase on the bottom is going to be doing the leading replication. It simply attaches nucleotides in position and moves down. You'll notice it found a T here. It put an A nucleotide with it because it needed the pyrimidine nucleotide that can make two hydrogen bonds. I'm sorry, the purine nucleotide that makes two hydrogen bonds to thymine. This is thymine here, which is a pyrimidine, and it can only form two hydrogen bonds. Similar rule here, except that this was the purine, so it put the pyrimidine thymine against it. Here it found cytosine, which is the triple hydrogen bonder, but this is a pyrimidine, so it put a purine on it. So it's going down the line, and you'll notice it's making an exact copy of what the other strand, of what the original strand was. Meanwhile, on the top, this polymerase started here and went to the left. This one's starting here, moving to the left. The next one's going to start here and move to the left. Those are Okazaki fragments that are being made. So as we move forward, you can see leading and lagging. Here's ligase coming in, bonding this Okazaki fragment to the first Okazaki fragment. Meanwhile, the polymerases just keep moving down the line. Ligases keep moving into position and bonding Okazaki fragments. What we have here now are two identical DNA molecules that are basically similar to what you see here in the bubbles. These are two identical DNA molecules that were manufactured by the copy process of the DNA polymerases moving down the line, along with the primases, the topoisomerases, the helicases, and everything else to put all these little nucleotides into position. So in this animated GIF, you can see that the same thing, that the nucleotides are actually being put in position, but in different directions on the two strands. Lastly, in this section, I want to mention retrovirus. Retroviruses are really an unusual biological phenomenon. Before we dive in to the next part, we told you at the very beginning of the unit that it basically is the case that living things generally use DNA as their hereditary chemical, and then they produce RNA, which then makes protein. Retroviruses are weird in that they store their biological information in RNA, and they possess a very unusual enzyme that's really important now to biotechnology. The enzyme's called reverse transcriptase, and this enzyme can make DNA from RNA. Now, this is really weird. Humans, as far as we know, do not do this. Most eukaryotes, or if not all, cannot do this. And most prokaryotes cannot do this. This virus can. HIV is a retrovirus. And basically, it and other viruses inject their RNA into cells. The RNA is, along with the reverse transcriptase that the virus injects, the RNA then makes a piece of DNA, double-stranded DNA. So this is the virus here. This is a HIV virus. It's sticking to a white blood cell. It injects RNA, that's this. The viral RNA with reverse transcriptase is used to make double-stranded DNA. This is the double-stranded DNA. That then goes into the nucleus of the white blood cell and literally splices in to a chromosome of the white blood cell. So the viral DNA is actually integrated as a loop, and it's put into a linear chromosome. The loop is open and it's literally stuck in. Now, what the virus then does is it uses that DNA that's in the nucleus to then make viral RNA, which makes viral proteins to make more AIDS viruses, which then, in waves of replication, move out of the infected white blood cells and to infect other white blood cells. And that's how AIDS is fatal. So HIV is a very strange virus. It's a virus that stores its information in RNA. It is capable of making double-stranded DNA from the RNA, and it uses that strategy to take over human immune cells and replicate itself. Now, the next topic we have to go into is basically gene expression.